background?
Yes. Yeah, we're going to ask you all about your research,
Sure.
I think.
Great.
We want to know how birds talk to each other.
Yeah, I'm happy to talk about that.
Anyway, go ahead and give me an introduction.
Yeah, so I'm Chris Thompson, or Dr. Chris Thompson on YouTube. I am a professor of neuroscience at Virginia Tech, and I study how hormones shape brain circuits, basically. I study it currently in a developmental animal model system. So I look at how thyroid hormone shapes neural circuits and neurogenesis in Xenopus laevis tadpoles, which is also known as the African clawed frog. I have a lot of experience with a bunch of different animals, including most extensively with a variety of species of songbirds. So songbirds learn how to sing, and that singing is regulated by a series of discrete brain networks known as the song control system. And I studied how hormones shape plasticity and neurogenesis there. So those are some of the main topics that I touch on. So, and yeah, happy to be here. I'm glad you had me on.
Okay. So you study embryology.
Yeah, so you could say that. It's a little bit later embryology, but that's where more of my focus is now.
Right.
Initially, I had started my PhD, so all my PhD work was on adult neurogenesis, which is an interesting topic in its own. So we're kind of related to neuroplasticity.
Yeah.
But now I've moved a little bit further up in the age timeline, and I focus more on early developmental events, primarily on how neurons get put into the brain. So the division of neurons in the brain and how they differentiate into neurons.
Yeah.
And and I've studied how thyroid hormone regulates that process. And I'm also using the tadpole as an animal model to examine how certain environmental contaminants interfere with thyroid hormone signaling, so kind of endocrine disruption type
Yeah.
mechanisms, and how that leads to diminished thyroid hormone dependent events in brain development.
Okay.
Yeah.
So let's see. I got into neuroscience
Mhm.
in about 2000 through evolutionary psychology is what I was really into.
Yeah.
And I was, Tubi and Cosmides and all the just so stories they came up with. And one of them was the 0.70 waist to hip ratio
Right.
preference of
The
males.
golden
Right?
ratio, sometimes
And,
referred
and
to.
and I asked myself, how in the hell does a brain wire an instinct like that?
Mhm.
And I wanted to know exactly how the brain. So I got into neuroscience, and
Mhm.
then I got into consciousness,
Mhm.
and I did about 55,000 posts on Richard Dawkins' forum until he closed it down, and another
Wow.
forum called Ratskep on consciousness.
Mhm.
And that's kind of what I got into, and at the same time learning neuroscience. And I posted it in private chat, but I got into a guy called O'Reilly and the hippocampus. And of course, I'm just a hack when it comes to neuroscience, but I was interested in how your daily journey gets stored in the hippocampus,
Oh yeah. Right.
geographically mapped, and then integrates with the cortex probably while you sleep. And O'Reilly is kind of into that.
Yeah.
So what is the current state of that? Has that become
Yeah,
an
that's
accepted
still kind of the
hypothesis?
larger organizing model. One thing that's fascinating about the hippocampal field is that there's this. Okay, we have what are referred to as place cells in the hippocampus. These are
Yeah,
cells that when a rodent is navigating a
yeah,
maze, a neuron might fire when that rodent is at a particular place. The hence
right.
place cell. And so we find these neurons in the hippocampus. There are other kinds of neurons you can find in the hippocampus too. And if you can record from enough of these neurons, you can kind of find a sequence of neurons that will fire as they're going along. We have now multi-unit electrodes that we can record
Yeah,
from multiple neurons at the same time. So we can watch as a rodent is going along this map or maze, the neurons as they're firing along, and you can organize them in that arrangement. Now, one cool thing that is pretty fascinating about this is that as a rodent is beginning its to start the maze, they will fire all of these neurons relatively quickly in the order that the neurons will fire as it's going along the map.
pre-planning. Yeah.
Yeah, pre-planning. So this is like what's referred to as a sharp wave ripple. So all of them fire with very, very fast. Within about 50 milliseconds, you get them all firing. So basically, the rodent, and obviously it knows the map at this point, right?
Yeah.
It's learned the map, and so, but it knows that hey, there's going to be some sort of treat at the end, say some cheese, and then the other thing is that when the rodent finishes the map, the neurons fire again, but in reverse order. So the last neuron fires first, and
Right.
then the first neuron at the beginning of the map fires last. And it does a reversed sharp wave ripple very, very
Yeah.
fast. So kind of like saying, hey, we finished the map. Let's make sure that we reinforce the circuit next time.
Right.
Yeah. And the hippocampus is probably what it's doing is that it's an experiential, so it's not memorizing the map per se. What it's memorizing is a series of experiences. So as you're going through the map, as you're going through the maze, you know that, okay, travel along a little bit, and I'm going to have to turn left, and now I'm going to go and turn another left, and then maybe right, and then left, and then back around. And so it's that order of experiences. So it's experiential learning that's being transposed in the hippocampus, and so that also applies to any other set of experiences. So it's not like so obviously it's important for spatial memory, but really what's happening is that it's a sequence of experiences that it's expecting to see, and that's the map.
Didn't they find that when the rat was sleeping that they went over the same map?
Yes. Then the sequences do the same thing. So you'll find the place cells do the same thing, but it tends to be faster than if it's actually navigating the map. And you'll see these similar sort of sharp wave ripples that are happening too.
Right.
Yeah. So it does the same. And the fascinating thing with songbirds is that we also can have, you can look at neurons within certain brain regions that are important for singing. And instead of having neurons that tend to fire a bunch, like they will in place cells in hippocampus, so spike rate will go up and it'll be firing a bunch and then it kind of stops. There's not like a code there. It's not like the sequence of the neurons, the sequence of the action potentials is particularly important. It's just firing a bunch if a rat is staying in a particular place. And then as it moves away, then the rate of firing is going to go down. That's like how a place cell works. And a lot of neurons seem to work that way. In the song control system, so this is this series of discrete brain nuclei that regulates singing, you can find neurons that will fire at a single point within a multi-second song, and that's it. And it'll fire a bunch within like 5 milliseconds. And so every single time the bird sings, it will always fire within that 5 milliseconds. And then you go to a different neuron, and then it's going in that same brain area, and then it's going to have a different firing rate. It's going to fire probably, again, 3 or 4 times within 5 milliseconds, but in a different place within the song. Because all the neurons have to work together to create the song. So you have that. You got a place code. And so that's one of the few behavioral examples that we know of having that level of specificity for neuron firing that seems to be associated with behavior out of all the animals that we've looked at. Okay. Now, the amazing thing is if you leave these electrodes in their brains and you let them fall asleep, you can see the same firing happen. So they also are dreaming of singing, it seems.
Right.
So probably the same kind of thing, and the importance of sleep, which is to help reinforce these, the firing and the pattern, so that the bird will learn it better the next day. So it's probably a memorization thing.
Yeah, I came up with a controversial disbelief in dreams. The fact that we dream, because I found myself recreating my dream narrative while I was waking up. So I think what was happening is, I've got all this stuff firing in my hippocampus, my cortex, that makes no sense at all. And then as I wake up, whatever's residual, I start to make sense out of it and come up with a dream narrative. So I asked myself the question, do we really dream while we're dreaming, or are we just like random garbage flying around?
Yeah.
But my son and I were on a road trip, and we got talking about the hippocampus and O'Reilly and the research and stuff. And I asked him, okay, let's recount what we did today. And what you do is you find the first spot and then the second spot. And so humans do with time what mice do with space. And of course, humans do it with space too. So I came up with another cute little narrative that the reason that we are as intelligent as we are is because squirrels used to have to know where they stored their nuts,
Mhm.
right?
Right.
so,
Yeah.
so little tiny rodents develop this, the allocortex and that
A
whole
very
system,
elaborate way of caching food so that they can find the food later on. Yeah,
right?
right.
Yeah.
Exactly. Yeah.
So we owe it all the squirrels looking for their nuts. So anyway,
That's
I don't
funny.
know. Yeah.
Yeah. Yeah. Animals have needed to know where they have go within space for millennia. And without a doubt, this is something that a long time ago was important. And so it's not surprising that like we have hippocampal structures, of course, in all mammals like that follow the same basic overall layout. When we
Right.
look at non-mammalian animals, so birds and reptiles, they also have a hippocampal like structure that seems to have some of the same basic connectivity. It's just not organized in the same way as it is in in mammals, but it does the same basic function.
Yeah, they're kind of like alien to us quite a bit. Their brain structure, right? Similar.
There's obviously less that we know about non-mammalian brains than we do about mammalian brains.
Mhm.
More than 90% of all research in neuroscience, especially today, is done just with rodent models, and most of that
Yeah,
is just on mice.
yeah,
So there's very little work that gets done with non-mammalian models for neuroscience. But that's not to say that zero. There's a fair amount that we do know. um yeah, and I mean, for I would say certainly over the last 30 and 40 years, uh I mean, maybe this is my bias coming out because I'm particularly fond of songbirds, but our understanding of non-mammalian brains, a lot of it has been filled in from the work that has been done with songbirds,
yeah,
and because of the song control system that I had the good fortune of studying for about 13 years or so.
yeah.
Mm.
So the system that creates the song in a songbird,
Mhm.
does that wire up in the embryo, or does it's obviously a combination? We got the nature
Yeah,
nurture false
sure.
dichotomy, but so the neurons wire into certain areas, and then that there's a proclivity
Yeah, they're
to.
born in certain regions, okay, and then they migrate and then organize in these regions. Um, then the connectivity that occurs, the wirings, so to speak, is something that happens during development. Some of that is being organized. So, okay, birds obviously are born in eggs, or at least they're developing eggs, and then there's post hatch um, that occurs. Most of the song control system is barely present at hatch. They
Okay.
still have a lot of development of their brain that has to happen at hatch. So,
Okay.
but I'm sure you've seen little baby birds. They look like gross pink, like a little rodent, right? Except they have a
Right.
little bit
Yeah.
of a beak. They're pink. They're very tiny. They're very helpless. So there's a lot of development that it still occurs, including a lot of neurogenesis. So that's the division and maturation of new neurons during those early few weeks. Then the song control system really starts to fully kind of get wired up. So if I'm talking about the species that we know the most about, which is the zebrafinch, probably are between ages 20 days post hatch to 35 days post hatch is when there's a huge amount of wiring and development that occurs
Right.
between the different brain areas that are important for regulating singing behavior. And then it continues to develop. And then, part of what I studied as a PhD student when I was at the University of Washington in Seattle was that these brain circuits are quite malleable. They actually change across the seasons in many species, especially those that have a seasonally breeding pattern to their reproduction and singing
Okay.
behavior. And so there's extraordinary change that occurs within the song control system, even as adults. And some of it is recapitulating some of those same juvenile mechanisms that occur when the song system is developing. So like it regresses and then grows again and regresses and grows again.
Okay.
Um, yeah, so it never fully ends in some
Yeah.
ways.
So when they're little tiny fetus-like looking birds, and they start to breathe or move, and they make a chirp.
Yep.
Okay. There's an audio feedback coming from that
Sure.
chirp, basically. Right. So there's like a feedback loop, and I think that applies to human development as well. Okay.
I'm going to close that blind. It's getting oppressive.
Yeah,
so yes, there's a feedback.
You had little ripples, and they kind of look cool. So I
did. Yeah, it was. Yeah. But now it's getting hot. So I often at this time, this window, I have to close it, and then I can't see the beautiful view outside my window. I actually have a nice view of the Appalachian Mountains here. But
there's this. Okay, I'm an embodied mind kind of guy for theories of consciousness. Johnson and who was it, Lakoff and Varela, Thompson and Varela. Anyway, so there's this, like when you move, okay, there's a feedback from your basically all your muscles and joint feedback, proprioceptive feedback. So the brain, so basically audio development is the same way. There's there's feedback through.
Absolutely.
And I'm guessing that's how the bird song is constructed.
It is.
But how much of an influence is culture, being with other birds that sing?
It's very important. So if you isolate a bird, and if it never hears any normal song, typically that bird will sing, but it'll be kind of mostly garbage. So they have to hear, so they have to hear a good song. And then if you deafen the bird, then it's almost unrecognizable even as bird song. So at least an isolated bird, it will sing, but it will not sound like a typical conspecific song. It'll sound really pretty messed up. But if it's deafened, now it can't even hear itself sing, and it is complete garbage. It's hardly recognized. It just sounds like squeaking noises. It hardly sounds anything like a bird song. Now you can also you can provide an audio tape where there's no social interaction, and birds will learn those songs. But they will learn better if there's some interaction with other birds. Primarily, it has to be a conspecific. There's examples of birds that will learn the songs of other species, including zebra finches and Bengalese finches. There's an interesting phenomenon of that if you give a baby zebra finch the choice between a Bengalese finch and a zebra finch, it's going to always pick the zebra finch to copy that song. But if it only hears a Bengalese finch, then it can pick up a Bengalese finch song and do a pretty decent job copying that.
Yeah. And there's a parallel in human development. And there, are you familiar with Marvin Minsky?
Yeah, Minsky, sure.
Yeah, and have you looked at his books?
I confess, I haven't looked at his books. I'm just familiar.
Okay, his books are incredible because he talks about the development of spatial reasoning and the early development of logic, like understanding that you could build a bridge with 2 blocks and all of this stuff about space and basically it's the same kind of feedback network. And he talks about the very, very rudimentary development of concepts that we get and how we place ourselves in time and space as, beings and how all that stuff wires up. And, I go on about, spiritual experiences basically are a meltdown of the temporal parietal lobe, which places you in space and time and as a thing and whatever.
It's where we have our sense of our body representation,
where we are, what we are, what we're going to do next. And so that relates to spirituality in a weird way, because if you have a grand mal spiritual experience, which is also another name for that is a nervous breakdown or maybe an epileptic seizure, basically all of these everything comes into flux. Now you were talking about these birds, how they melt down their pattern of song and rebuild it again. So these birds are having a spiritual experience. They're basically melting, because that's what happens to people. You can melt down your belief system and you can completely change a lot of core beliefs depending on what you run into next.
This is
I had this happen to me. That's why
Yes. So
I haven't
I
seen
will not call this
it.
seasonal change a spiritual experience.
You should. You should.
You could make the metaphor, and I can see that. I mean, that's why like we have bunnies and eggs as part of our Easter festivities because it's part of
Right.
rejuvenation, right? And that
Right.
you know, things around nowadays, right now, like with Halloween and death and loss
Yeah.
as the law, you know, like what happens at the end. So we certainly make these connections to the seasonal pattern that occurs. So okay, so one of the, I think the amazing things about songbirds is that there are more songbird species than there are mammals, like all the mammals.
Really.
So yeah, just a a few hundred more, and so that doesn't include all the other birds, right? The penguins and the
Right.
gulls and all of them. It's if we're just talking about songbirds, so there's a huge range of variation that you can find just within songbirds. Some live in tropical areas. Many live, you know, many live in tropical areas. Many live in temperate areas. They live all over the place. And then the kinds of behaviors that you can find. There are most species, it's going to be just the male that sings, and the audience is oftentimes the female, but sometimes it's other males because it's
Yeah.
used as a way to communicate like where we're located in space, or like if I have a territory, I am here and my
Yeah.
neighbor is over there and I know that that's my neighbor, and so if I hear his songs over there, I'm okay with that. But if I hear another bird song over there, well, he might be some broski coming in to, you know, hone in on my mate, so I need to go over there and kick his butt. That's essentially that kind of communication that occurs, and they'll learn. So obviously, they have to learn like spatially the repertoire of some birds versus others. So, but then the seasonal aspect is that some birds will learn their song, and then they will have a degradation of the song control system, and then it just rejuvenates in the next spring. But that song doesn't really change much from season to season. There are other birds, like canaries, for instance, who go. This process of seasonal change seems to be really critical for unlocking their ability to incorporate new songs. So when they are breeding, their song tends to be quite stable, and they don't really learn new elements to their song. But in the fall, when they're not breeding, the song system starts to break down a little, and it probably is unlocking some synaptic availability
Yeah.
in the song control system so that they're a little bit more free to incorporate new elements. And so they're much more likely to to introduce and learn new song elements in the fall. And then the next spring, now they have a new song or new song elements. Canaries tend to be a bit variable in the kinds of songs that they sing. They will be stereotyped in the notes that they sing, but the number of times that they sing individual notes can vary. It also depends on the breed. I'm getting a little sidetracked with the details,
That's
but
yeah. That's interesting that there would be this meltdown aspect.
yeah,
You know,
right,
basically synaptic meltdown.
sure.
Uh,
Some people have referred
humans should do that between elections.
yeah, I know, right.
Have a complete meltdown and
There,
rethink the whole thing,
yeah,
right?
yeah.
Yeah.
There definitely appears to be like a. I mean, I think we're seeing a constant meltdown and no rebuilding in at least in some aspects of our political electorate.
Something's going wrong.
Yeah,
Something is really wrong.
right. Well, that
Yeah.
honestly, I don't. I know you're very well read. I would guess that you're familiar with the lead crime hypothesis.
The what?
The lead crime hypothesis.
No.
Oh, you're
Oh,
not. Okay. So
no.
this is a something that I think may be affecting the boomer generation more than people give appreciation to. So one of the toxicants I'm working on is lead at the moment. So I'm looking at how lead, well, I'm testing a hypothesis on how lead impairs brain development.
Okay.
We know that lead can impair neuron function in many ways, and that's likely to be very true
Yeah.
in the
Yeah.
developing brain. But one thing that I think is largely understudied is how it affects how thyroid hormone gets into the developing brain. So you have to have sufficient levels of thyroid hormone getting into the developing brain early on in development, because if you don't, you end up with microcephaly. The brain is too small You
Right.
haven't had enough neurons that got born, and its most severe case, it results in cretinism. So the term cretin comes from that condition, which used to be a lot more common in our society than it is nowadays, because we test for thyroid hormone very early on in pregnancy, and we have pretty easy ways of treating women who have, say, insufficient levels of thyroid hormone. So, okay, I'm testing how lead impairs how thyroid hormone gets into the developing brain by affecting the distributed proteins that carry thyroid hormone through our blood and into the brain.
Okay.
Okay, so all that's kind of like where my interest in lead comes from. But one thing I've learned about, and one of the things that got me very interested in looking at how lead affects the developing brain is actually, I got to give credit to a particular writer he was the main blogger for Mother Jones for a very long time. His name is Kevin Drum, and he's written a number of articles for Mother Jones, and it's worth looking these up because they're really fascinating on the lead crime hypothesis. So
Okay.
what Kevin so I can would it be possible for me to share some slides,
Yes.
because I actually just talked about this.
Yeah. Do you know how to share a screen on
Yeah,
StreamYard? Okay.
I don't. You'll have to walk. I think it says, I see
Okay. Go
present.
down to present, that little
Okay,
button at the bottom.
that'll do it.
And then you got a choice between a Chrome tab or,
Okay, or a PowerPoint tab. All
you
right,
try.
I'll make
But
this work.
yeah, when I was a kid, they used to give us mercury to play with, and we'd rub it on pennies,
Oh,
and
okay,
make them silver, right? Mercury, so we were rubbing mercury, and of course the lead
right.
paint was like in every house, it's like, go chew on some lead paint, you know,
Oh,
go
funny.
play with the paint. Yeah,
Okay,
but
so okay, I'm going to
hopefully
share my screen.
okay.
This will work better, and because I can't upload. Okay, so hopefully you can see this. Yes, there we are. Good.
There we go.
All right.
Oh my God.
So I wasn't planning on doing a lecture, but I promise you I won't try to overtake it too much.
No,
But
we
this
can do
is
anything you want.
cool. Awesome. So this is some interesting stuff. All right. So the lead, so okay, one thing that we know about lead is that it leads to cognitive impairment. And so what we have here on the x-axis is concurrent blood lead levels. So micrograms per deciliter is typically how we measure lead in blood. And so this is a combination of a series of studies looking at the correlation of IQ and blood lead levels, and you can see that there's a significant drop with relatively low concentrations of blood lead levels. So 5
Right.
micrograms per deciliter is generally considered to be like the important cutoff. And then 10 micrograms per deciliter. Well, the EPA recently moved it actually even less than 5, but it was from 10 to 5. Now they say that 3.5 micrograms per deciliter is like the cutoff. But you can see there's like at least a 5-point IQ drop with less than 10 micrograms per deciliter, and it just gets worse as blood lead levels go up. So
Wow.
there's that. Obviously, lead kind of got into the news with the Flint,
Yeah.
Michigan water crisis, and this was just a pretty stark example of that. We still have dormant sources of lead that can be a major issue to populations. So what happened in Flint was that the Flint government got taken over by the state government. They appointed an administrator. They got rid of all of their politicians. And then that administrator started making decisions to just try to save money. And one of the ways that they decided to save money is that they stopped buying water from Detroit, and they instead were taking water from the Flint River, which was so highly polluted that even the corporations, the car manufacturing companies along the Flint River, they refused to use the Flint River because they knew how contaminated it was, even though
Yeah.
they're probably the ones that contaminated it. So they wouldn't even use it. But here now Flint is saying, hey, why don't we use just the Flint River, because that'll be a lot cheaper than bringing in water from Detroit. So this is Mark Edwards. He's a professor here at Virginia Tech. He was core to like uncovering the Flint water crisis and identifying that there was huge amounts of lead that was being released from the pipes because the Flint River was corroding those lead pipes. So generally, a lead pipe system will be okay as long as you put in corrosion control measures to make sure that the pipes don't get corroded. And that was one of the things that the Flint administrator decided is like, we're not going to spend money on that. It costs money to prevent
Right.
corrosion control. So then this is what the water looked like. So this is zoomed in here in Flint water, and you can see how it does not look like
Wow.
something you would want to drink. Yeah,
No.
awful stuff. So this is the thing about water and lead is that cause for concern, the level that we say is cause for concern is 5 parts per billion. So that's 5 lead molecules per billion molecules total. In Flint, the average that was when Virginia Tech went there, they took hundreds of samples, and they found that the average was 27 parts per billion at the peak of the water crisis. And it went on the highest level that they could find was 158 parts per billion. So quite a bit more than the cause of concern. And then in one other study that was not done by Virginia Tech, they found as much as 13,000 parts per billion. That's a lot. That's a lot above 5 parts per billion. So one thing about then is that, so this is taken from a Reuters article, really great article, looking at lead levels in children. And so this, what we're seeing here is a heat map of Flint. You can see that somewhere around 10 to 15% of children were testing for elevated blood lead levels, more than 5 micrograms per deciliter. Okay, so there's that. Now the thing is, the Flint water crisis certainly showed us that there's a problem with lead, but it vastly understated the problem. The problem is actually really across the United States. So this is Flint here. This is Detroit. Here's Jackson, Kalamazoo, and Grand Rapids. And you can see they all have also elevated blood lead levels in children. And in fact, Detroit, you can see it's particularly bad. 25, 30% of children in Detroit with blood lead levels that are relatively high. This isn't just about Michigan. That you can find this in a lot of cities. So this is Baltimore. You can see some areas the children have more than 40% elevated blood lead levels. Cleveland is really bad. Milwaukee. This is true for just about every largely older industrialized city in the Midwest and in the East Coast that you're going to find.
Got one for Minneapolis.
I'm sure that's in the map. I would guess that it's also equally high. And the reason why is because there's still lead contamination from other sources, not just drinking water.
Yeah.
So this is the thing. This is about the lead crime hypothesis. So lead. So here, we're zooming in here, and this is actually taken from Kevin Drum. So I'm giving him credit from. He's no longer with Mother Jones, but he was with them for a long time. So what we're looking at here is a line for 5. This is micrograms per deciliter is the way that he put it. And then we had some children that had above 10 micrograms per deciliter. You can see that Flint switched from they were taking water from the Flint River, and you can see that blood lead levels increased. And then when they switched back to Detroit water after the crisis, blood lead levels went down. So evidence that it went up and then it went down. And but the thing is, this is the historical data for lead poisoning in Flint. You can see that in the late 90s and the early 2000s, nearly 50% of kids had more than 5 micrograms per deciliter. And quite a few of them had more than 10 micrograms per deciliter. This is because we have other sources of lead poisoning. One of those is, of course, lead pipes, but another is lead paint, which is still a problem. We have lead paint that had been used from the 20s up until the 60s, still in many homes, particularly in poor urban areas. But another thing that's largely appreciated is that we still are dealing with lead contamination from when we added ethyl lead to gasoline to make leaded gasoline, right? So we added that to prevent cars from knocking and causing all kinds of noise. It also added power, as you can see from this, fun little graphic, to making, somehow advertising leaded gasoline. But the thing is, it was just dumping out lead in particulate form as an exhaust, and then it would settle down because it's lead, right? It's not going to hang in the air for too long, and then contaminate soils. And so this is work from Howard Milkey. He's a professor at Tulane University in New Orleans, and this is a map of New Orleans. And so what we're looking at here is lead concentrations in soils in many neighborhoods throughout New Orleans, particularly the poor black areas like the lower Ninth Ward. This is the Mississippi River going through New Orleans, and you can see what we're looking at here is lead concentrations in soils in milligrams per kilogram. So what's considered safe for children is soils have to be less than 400 milligrams per kilogram. And you can see many of these areas have a lot more than that. And some areas have as much as 1,700 milligrams per kilogram. The other thing that Howard did is that he measured, so he was looking for a relationship of soil, and then he also measured blood levels for children that lived in those areas. And you can see 5 micrograms per deciliter, that's right here. And there's a lot of kids that had more than that. And it's not a nonlinear relationship, but you can see that there's a relationship of more lead in the soil is correlated with increased blood levels. And it's in the soil because, and this is data that he collected prior to Katrina. That was 2005 when Katrina happened. I think these were data taken in 2003, and or actually it was over a number of years. But he what was I saying? Oh, this is about 20, 30, maybe even 40 years, about 30 years after lead had been taken out of the soils. And so, lead had been taken out of fuels. So it's a persistent problem, and this is probably what's causing problems in many of these urban areas.
And cleaning it up would cost so much money that nobody wants to address that. So what is the biochemical pathway for
lead? Let me tell you about the lead crime hypothesis real quick, because I'm almost there. Okay. So we have the cognitive effects. This is likely to be impairing how brains grow and develop. Now, the thing that's interesting is that, okay, I was showing you percent of children above 5 micrograms per deciliter. What we know, though, is that in the United States, so this is looking at average blood levels in the United States. So the average blood levels of toddlers in the United States between the middle of the 1950s up until the middle of the 1970s was greater than 15 micrograms per deciliter, and it peaked at the late 60s and early 70s greater than 20 micrograms per deciliter, and it got up to around 23 micrograms per deciliter. Remember, that's the average. So that we're not talking about percent of children above 5 micrograms per deciliter. Probably every single toddler in the United States had more than 5 micrograms per deciliter at this peak for this to be the average. There probably were toddlers that had 40, 50, 60 micrograms per deciliter, stuff that we know that can actually be toxic to adults. But the average was around 22, which is already correlated with a distinct decrease in IQ, right? So this is IQ. Yeah. And you can imagine all kinds of other behavioral effects. So the other thing that's interesting about this, and this is what Kevin Drum says, is that we had this late 20th century crime wave that occurred. So this is number of violent crimes per 100,000 residents, I think, per year. And there was what we have on the green line is this is the years for the lead data. And then the blue line, this is the years for violent crime. And what Kevin Drum did is he just moved it by 23 years. So if you were a child that got poisoned with lead, now there's a percentage of them that have been pushed to be more impulsive, aggressive, and likely low IQ. And that's going to lead to maybe a greater propensity for committing certain violent crimes. And then when lead was pulled out in the late 60s and early 70s, well, about 23 years later, we all of a sudden started to see a decrease in violent crime in the United States. And it's only continued to decrease since this has been made. It's only gone up a little bit in the last few years, but it's still well below what it was in the early 90s. And we don't have a good reason to explain why that is. Giuliani, he wanted to say, "Oh, it's because I did the what was it? His anti-crime measures, the glass, broken glass measures, and stop and frisk, right? But that's one city. This is across the entire United States. We saw this phenomenon. Yeah. And how else can you explain it? I mean, it's an entirely societal effect. Um, and this kind of relationship has been shown in other countries. So the United States was, believe it or not, one of the first to take lead out of gasoline. There are others that were delayed in that. So in Europe, there was a crime wave that occurred that only abated once like 20 years passed when lead was taken out. So it's likely that the kids who were born in the 40s, 50s, and 60s were heavily poisoned with lead.
I was born in 51.
Well, there you go.
Yeah, that's why
You might have just beat it. Like, right.
I
So
can
you're
never
right here.
remember what I can never remember what the superior colliculus is.
Yeah. Oh, so there you go. And that's actually one of my favorite brain areas.
Yeah, that's probably
Yeah.
because of the lead.
Yeah. Yeah.
So, yeah.
So, all right, I'm gonna go back to.
Well,
So yeah, that's the lead crime hypothesis. And if anyone's interested in that, I highly encourage you to just search Kevin Drum, DURM, lead and crime, and you'll find two really good articles that he's written. He also, as I said, he was the blogger for Mother Jones, and there's a bunch of interesting blog posts. I have to admit, like, that was the inspiration for me even getting interested in the topic. And while I'm doing research on it, and now I've just sent in a grant to the National Institutes of Health to address this. So
what does a lead do by physiologically?
So lead shares similar electron valence to calcium, and calcium is very important for all kinds of molecular pathways, particularly in neurons, right? Calcium is the main thing that allows for neurotransmitters to be released, for instance. So, but it's involved in a lot of things within a cell. So lead probably gets into cells and interferes with how calcium functions. And so that's likely to be some of the explanation for why lead toxicity affects neuron function. But like I said, I have this other, there we are. Yeah. America's Real Criminal Element.
Yeah, interesting. Okay. Yeah, I'll link that when we premiere this. So
sure. Cool.
Yeah, so if it affects calcium is so damn important for synaptic function.
Exactly. Right.
And development. Yeah, well, that's going to be my go-to excuse from now on if I get pulled over for going over the center line.
Like, hey, but honestly, I think there's, these people born and the thing is, it's a shift in the distribution, right? This is the thing that gets lost in some of this. It's not like every single person is going to be equally affected in the same exact way, that they're all below 100 IQ. Obviously, that's not the case.
Right.
But if there's a shift in the distribution, you have a tail end that's now going to be more, say, prone for aggressive behavior. Some of them are going to end up committing violent crimes, particularly when they're young and stupid. But the thing is, some of that aggressiveness is going to persist. And the thing is, like, these are the baby boomers, and they're the ones who are kind of in charge now. And so you could just imagine what that shift means for the basic population. Like, they're our bosses, they're the managers, they're the politicians. Lack of empathy, loss of sense of community.
Old people really piss me off.
And it's not a universal old person thing. But I think there really is something to be said that, like, there's a generation that probably got hit, and not just with lead. I'm sure there are other things that they were exposed to. And I honestly think that there are other things that we're exposing kids to at least 10 years ago and 15 years ago that we're starting to see the result of now. Like the sharp increase that we've seen in autism. Not all of it can be explained with just increased diagnosis. There's some of it that can be, but it's not exclusively that. There's definitely other environmental impacts. I have some ideas about what that is. Not to say that that's the whole thing or explains everything about it, but and there's other research. So in fact, I'm working on another chemical that's sort of related to that topic.
Which one? Do you want to tell me?
Let's see. I guess I could say. I mean, it's not like it's based on research that's already published, so I can talk about other people's research. So there's an antimicrobial agent known as triclosan, which had been widely used in all kinds of consumer health products for decades, and its use only increased in the 90s and 2000s. where it was found in many, 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 many products, to the point where it became a major environmental contaminant, where it was like one of the top 10 contaminants that you could find in streams and rivers near urban areas. And it's also very difficult to remove out of wastewater treatment. Like you have to do active removal, otherwise it passes into, like, if you're going to reclaim water. Oh, look at that kitty! Holy cow!
It's a big one. I had to move them.
Kitty alert. So it's an antimicrobial agent. It's used to just prevent spoilage, primarily in, and in some cases to treat certain things. So Colgate Total, for many years, had triclosan in it. That's the only reason why it was called Colgate Total. And but they had a very specific medical claim, and so you would know that triclosan was in it. The claim for Colgate Total was that it reduced gingivitis, which is caused by bacteria, right? So it makes sense that you put an antimicrobial agent on your teeth; it's going to prevent gingivitis.
Yep.
But it was in lots of other products: soap, deodorants, mouthwash, cosmetics, also clothing. Yeah, there's triclosan. It's got 3 chlorides.
Yeah, it's
guys referred to as
anything with like every time I look up a medication that's got chlorine sticking out of it, I always get scared.
Yeah, it's a halogenated diphenyl ether is what
Yep.
you can refer to that chemical as.
Yeah,
And
it
so
is.
and it's similar to thyroid hormone in some ways, which is why it came across my radar. But I don't believe that it's I don't think that there's good evidence that it's actually a thyroid hormone endocrine disruptor. However, there's more recent evidence that shows that it impairs mitochondrial function. And so I have some data coming out that'll hopefully be out soon, where it looks like it may interfere with how thyroid hormone works in the brain, but not through classic endocrine disruption dysregulation, but instead is impairing how mitochondria work. So, and there's a bunch of studies out now that show that. And then there's one study that was published in, I think this was in the prestigious Journal of Nature. I think it may have been Nature Neuroscience, now that I think about it. As our friend might say. But this study, what they did is they gave pregnant rats triclosan, and then the baby pups were born. And then the rats were still continuing to give triclosan through up to weaning, which only takes about 20 days in rats. And then they raised those rats up to be adults, the babies. Then they did a whole bunch of social tests on them and found that they have some really significant effects on their social affiliability. So they tend to avoid nestmates. I think that's what it was. But they also are uninterested in strangers. And then they have an obsessive grooming that they do. So the grooming that they do is around 4 times more than what control rats will typically do. So the grooming is off the marks. And they have other behavioral issues. And these are adults, but they were exposed to this triclosan in early in development. So some of that has to be replicated, of course. But there's some reasons to think that. So they imply that triclosan may have led to social difficulties for these rats that's sort of autism-like. So that's a connection that's a lot more tenuous, I will say, than like the lead crime hypothesis. And even then that one's somewhat tenuous. There's still a lot of work that would need to be done to prove that or demonstrate it with some significant level of confidence. But
it'd be interesting to figure out what exactly is not wiring properly.
Right. And I think that it's mitochondria. I mean, it's definitely a very potent mitochondrial, the term is uncoupler. So it affects how mitochondria, the electron transport chain in mitochondria work. And so that gets all messed up. And I actually am working on that right now in my lab. We're focusing on how certain contaminants impair mitochondrial function in the developing brain.
Yep. Okay, so let's see. Let's switch to creationists.
Yeah, let's talk. That's what brought us together.
Yeah. So did Cell Cordova mention the protein evolution thing, the common ancestor of proteins?
and
Yes.
it's such a bogus argument. I mean,
Well,
I
yeah.
okay, I want because I heard your review of our debate, and I agree with a lot of what you had to say.
Yeah.
Now, Aaron and I talked before. Obviously, we had a strategy, and
Right.
our strategy was to make them talk about the sillier aspects of young Earth creationism. You know, we know
Yeah.
Sal loves this protein argument. He loves to talk about topoisomerases. I prepared a little for that, and I busted that out in my part of the debate. But we just mostly ignored it. It's a dumb argument. I mean, you actually laid that out really nicely in your review.
Well, yeah,
Um,
I'm
yeah,
not sure how well I did, but basically he's saying that it's a composition fallacy, right? He's saying that
it is.
because all animals derive from a common ancestor, then all
Right.
proteins had to too. But
Exactly.
when we look at prebiotic Earth here, okay, what we've got is we've got I hate to use the word soup, but I'm gonna like a
Yeah.
soup of basically RNAs. These are RNAs,
Yep. If
and
we're
these are
working on the RNA world hypothesis, which there's a
and
lot
these
of
are
good
many,
reason to think
many
that.
billions and
Yep.
trillions of small peptides.
Exactly.
And what happened is at some point, basically when membrane encapsulation became a thing, or however things were encapsulated, basically a cell would, take a number of different small peptides. The thing about small peptides is they can stabilize RNAs. So this is like a little RNA here.
Right.
This is a small peptide that gloms onto it and stabilizes it.
Mm-hmm.
So that kind of stuff was going on. And we get all of these cells basically that all have different, number of different peptides. And that is what evolved into all the different families of proteins.
Yeah.
Because one of these cell systems worked. One of these cell systems had the right metabolism. And there's so much more to abiogenesis. Like for one thing, if you have an alpha helix in a protein, it will embed itself in a membrane almost automatically.
Sure. Right. Yeah. Yeah.
We
It's a
we can do
membrane
that
target.
individually. Yeah.
Exactly.
Yeah.
It's one way you can look for membrane targeting. And
yeah, so you've got porosity in these cell membranes, and this whole thing. But anyway, so Cell's argument, and I don't know why he will not get over it. I've taken down his argument because he won't talk to me, right?
yeah.
So I can't address him directly. But he claims like Dr. Dan is the only one that agreed with him. That
I know. I've seen him talk about that clip. He takes like 10 words that Dr. Dan said, where he's like, well, yeah, that's right. Of course, there's no common ancestor for proteins. But it's like,
right.
Well, no one thinks that there was like a single
Nobody
common
thinks
ancestor
that.
for all proteins. Right. Like, that's not how it works.
It's just crazy. And then he goes on about zinc fingers, and he completely misrepresents zinc fingers. So, so I delved into
collagens too. Like the collagens, like they vary quite a bit across different organisms. Of course, there's like the underlying, what is it, the G that's in there that is every third. But there's a family of collagens, and the collagens actually do a bunch of different things. They're not just extracellular matrix there to sort of cushion cells. That's one thing that they do, but they're used in the brain as a signaling mechanism. And there's whole papers out there on evolution of collagens. Now, I it's not what I work on, so I haven't looked
I think there's only
much,
5 of the collagens that actually hook up and form polymers, and the rest of them are signaling. And the other thing for collagens and zinc fingers, what you're going to find is repeating domains.
Right.
And once you have 2 domains that are similar, okay, they're going to overlap occasionally, and you're going to end up with 4 domains, and they'll multiply. Yes. And zinc fingers, like he goes on about zinc fingers and how could that have evolved?
Right.
Well, zinc fingers are like, we have 700 of them. We know what about 100 of them do. The rest of them, I think, are just, they're sort of junk drawer DNA.
Mm.
And the interesting thing is, if we have like a transposon, an ERV, or, an ALU that jumps in the genome, if that thing would cause a problem, there could be a zinc finger in the library, ready to glom onto it, wrap it up in the histones, and get rid of it. And then that will end up, then that zinc finger and that ERV will end up possibly becoming fixed in a population.
Sure.
But what we have here is we have we basically have junk masking garbage, and
Right.
it gets fixed in the population. No
Sure.
function.
Right. No function whatsoever, but
pass it
it's
on.
there. But
And that's the way zinc fingers work. Yeah.
right. And then you
And
have to wonder, well, so it was designed that way, like
yeah, did you create something to kill us?
right.
And it jumped around, and then you created something to remedy the thing that was going to kill us. And
Mhm. No, none of that actually happened because it's just the sequence that currently does nothing in their
does
DNA,
nothing.
right? So,
Yeah.
and that's how it was designed. It was just made to look like these other things that we know have some function and how they work.
Right.
But no, instead, you would say that they were specially created that way, that the designer laid down the remnants of zinc fingers and ERVs in our DNA at some point, and they did this in all animals. And
they have. And then their narrative is that, well, we only know the function of maybe 20 or maybe 40 ERVs right now, right? Or transposons. But we're going to discover the function for those other 10 million
right
any day now, right?
It's
like,
sure.
No, not all of them are functional.
No.
Until they become functional, until they lock into a biochemical network. And that's kind of my big thing, is that we have this huge library of connections and networks that don't go anywhere right now.
Yes.
And every now and then one of those networks will lock in and it will do something. And now you have a beneficial mutation.
Yeah.
And that can become a new pathway into the future, or it can change how an embryo develops.
Yes.
And changing how an embryo develops is kind of like the tail wagging the dog.
Right.
A very, very small change in a transcription area
can have enormous consequences.
Will have an enormous change in bone structure. Or whatever.
There was a recent paper that came out from, one of the authors was Svante Pabo, who is the new Nobel Prize winner this year. This paper was in the prestigious journal Science, I believe.
Prestigious journal,
yes, very prestigious journal. And yes, by the Nobel Prize winner, Svante Pabo.
Yeah, you got to mention that.
That's what it matters. But it is cool. But Svante Pabo got the award for his work on ancient genomes, which I think is super great for this whole creation evolution debate at this moment. And I don't think that they've wrestled with the results at all in any respect whatsoever. And especially the genetic entropy argument, it's okay. If this is genuinely true, we have ancient DNA that we can look at. We can compare it to the human DNA. If you really believe that that DNA is degrading so rapidly, then why don't we compare sequences from, say, Cro-Magnon man or Neanderthals, which is what Svante Pabo got his award for, his work on the Neanderthal genome. If that should mean that they should be very, very similar back in if those were like the precursors to humans, like if those were daughters and sons of Adam and Eve, shouldn't they all be very similar? If Neanderthal and Cro-Magnon man were both sons and daughters of Adam and Eve, then but we know that they're not. We know that the Neanderthal genome is quite a bit different from the human genome. Wouldn't that mean that there was incredible entropy that already existed way back then, and that the human genome nowadays isn't even anything like the Neanderthal genome, except for, of course, Northern Europeans and also parts of Asia that have Denisovan DNA in them, right? Which is a different kind of Neanderthal ancestor. So this is all work that Svante Pabo got. Anyway, I was getting to his paper in the prestigious journal Science, where they just showed that there's one single mutation, an amino acid substitution in the human version of a gene that's expressed in neuroprogenitor cells, which divide into neurons, a mutation compared to the Neanderthal genome. And the Neanderthal genome looks more like other apes. So we have the single mutation that's different in humans. Neanderthals have the mutation that would make them look like the other apes. And this single gene, so we know that the human brain and the Neanderthal brain were similar in many ways, but there were some differences, particularly in the frontal and prefrontal cortex. Right. It tends to be larger in human brains than it is in Neanderthal brain. And I should say Homo sapiens versus Neanderthal. Um, uh, so okay, so you've got this difference in frontal lobe development. They took this gene and they mutated it in rats and in ferrets, and they saw much greater neurogenesis as well as even enhanced folding of the frontal and
Okay.
prefrontal areas in those
Yeah.
animals. Yeah. So
Yeah.
a single
You know, that,
mutation, right, can lead to this massive change. And obviously,
yeah.
that's a developmental event. It's something that happens early on, neurogenesis. But if you just do a single mutation in one gene, now you can lead to this, substantial increase in an entire part of the brain.
Yeah. 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 I think I read somewhere a long time ago, and I'd have to fact check this. There's part of the, I think it's a basal ganglia
Hmm.
on humans that actually swells into one of the ventricles.
Mm.
So it's a much bigger area, but humans are the only primates that have this.
Hmm.
And it looks like an extracortical loop that goes, between the thalamus, basal ganglia, and the cortex.
Hmm.
And I, yeah, have you ever heard anything about this?
That
Like,
that's specialized in humans and not in other mammals. I have not.
yeah,
Uh,
it's something like a new circuit or an added on circuit with the
sure.
frontal lobes.
Okay.
Basically, that would probably give us more control over our working memory.
Okay.
And uh,
Oh,
I,
that's interesting.
yeah,
Yeah.
I'm
So, I mean, there's
running
a
into that.
basic sort of pathway that goes through the basal ganglia. You have cortical input
Yeah,
to the striatum, which is one part of the basal ganglia. Then that goes inputs to the globus pallidus,
yeah,
and then that inputs onto the thalamus, and then the thalamus then returns that back up to other parts of the cortex.
yeah,
Oftentimes, especially if we're talking about the areas that are important for procedural motor memory, that's
yeah,
feeding into either premotor or motor areas to drive motor function. So that we can do like so the basal ganglia is very important for learning and memory, but usually the things that we have to practice and memorize how to do with our physical practice, like swinging a baseball bat or playing the piano. Those are all procedural memories
yeah,
that eventually you get so good you don't think about it. You just sit down and play the piano, and your hands are doing the things that they need to do because it's the basal ganglia that's taking over generally for
yeah,
those things. Now,
yeah,
I could imagine, given the expansive frontal and prefrontal areas in the human cortex relative to the other even apes, that maybe there's some additional connectivity into the basal ganglia that I'm just not aware of. I wouldn't be
yeah.
surprised if that's the case.
I'll see if I can find more about it.
Mhm.
I've
Sure.
got a 10 volume set on the basal ganglia,
Mhm.
which I
Oh, nice. Holy cow.
I had
It
tried to
is
read,
it's
but
it's a very important part of the brain, for
yeah,
people who I was gonna say, for people
it's
who have
about
problems with
5 feet.
they have an appreciation for it, right? So,
Yeah,
for people with Huntington's disease or Parkinson's disease,
yeah.
those are the areas that are mostly affected, the
Well,
basal
there's like
ganglia.
these action selection networks in
Mhm.
the that work in the basal ganglia. And
Right.
yeah,
The cool thing, to return it back to the songbirds, and this is one thing that I had thought about touching on in our debate, but Aaron honestly was on a roll, so I just let him
yeah,
continue to smash down
right.
arguments here and there. But that we have shared connectivity in all vertebrates for overall basic neural structure. Now, it's not identical in many
Yeah,
respects, but the connectivity, a lot of it's there, including a cortical basal ganglia thalamocortical loop that exists in the song control system. So this system that regulates singing behavior in songbirds, we see the same basic connectivity. The surprising thing was that at one point we didn't see the splitting of the striatum and the globus pallidus. Like, we saw a cortical-like structure connecting to an area that we thought was basically basal ganglia, and then that outputted to the thalamus. But shouldn't there be two parts? Because that's what it is in mammals. It turns out that in birds, it's just all mashed together. So they have striatum and globus pallidus neurons that are all just sort of mashed together.
yeah,
And so then we don't have anatomical separation, but connectivity wise, it still works that way. The cortical part connects to the striatal neurons, and then those striatal neurons connect to globus pallidus neurons, and those neurons then inhibit the thalamus, which it
yeah.
works the same way. And that's 320 million years of evolution separated between
Right.
humans and birds, and yet you still have the same basic connectivity, and you can see it in all vertebrates for the most part. you know. So,
Yep.
But no, they are all specially created in some factory. Yep.
So, do you get into consciousness much, or think about consciousness?
So that's not like, it's getting very far from my expertise for sure. Okay. But I have thoughts on consciousness. I don't know if, I think you saw a little bit of my discussion with Sal when we talked about consciousness. And I mean, I utterly believe that consciousness depends upon activity within neural networks. We can become unconscious in ways by diminishing activity in brain areas. When we are asleep, we're unconscious. And that you can't have consciousness without a brain. I mean, until I see otherwise, I think that there's possibility. Like, I could imagine with artificial intelligence that we may develop the ability for an alternative set of algorithms that might be extra neuronal. But I don't think that there's like some sort of unifying consciousness in the universe, which is what Sal was arguing for. And because it doesn't make a lot of sense to me.
I've been fighting that battle for about 20 years.
Hence the name Answers in Atheism. Which I assume that's supposed to be a take on Answers in Genesis, or
it is.
Okay, great.
Yeah, and it's sort of I'm a spiritual atheist, right?
Yeah,
because I understand that I'm an organism, and that I'm primarily driven by emotions and feelings in my body, and I'm a thing in the world, right? And I call that spirituality, basically connecting to the world and shutting off your mind. So anyway, I do have some answers in atheism. One of the answers in atheism, this thing about lead that you were talking about, and other things that affect the brain, we know so much about neuroscience right now that it should be altering the way we raise and teach our children, because we understand a great deal that we are meat brains, and there's shit we can do to make these meat brains work a little bit better in society. And until we get rid of this idea that it's a spirit mind, right? That there's nothing we can do about it, we're not going to progress as a culture, basically, and learn to use what we've learned in neuroscience. We've learned a lot about violence, in particularly in males, and the sources of violence and the sources of problems in human society, and that's part of Answers in Atheism. That if you take a perfectly naturalistic view of what we are, and you understand that we are just little blobs of goo on a speck of dust, and we get humble and start altering what we do with ourselves based on science. Until we get to that point, we're not going to progress, and that's kind of my thing. So, that's one of the reasons why I fight the religious people.
But
I don't want to throw the spirituality out the bathwater, because spirituality is cool.
There's also something that religious communities can bring, which is a sense of community and fellowship that needs to be replicated in other ways, that doesn't have to have the silly religious garbage aside it. But you can see that that's the thing that draws a lot of people to religion, is that sense of community and fellowship. And that makes sense to me. We're very social creatures, and a religious community can serve as a source for comfort and help in need, and just for friendship and fun. And obviously, like that, like, we still have to have some of that. It just doesn't have to have the religious baggage on it, which unfortunately too often then becomes a manipulative tool to take advantage of people.
Anytime you add dogma to the equation, you can have problems, basically, based on the dogma, that sort of thing. So, but in this atheist community on YouTube that we're a part of, And I met Arn Ra down in Texas a couple of times, and had him on the channel a few times. So there's Arn Ra, and there's Erica, and there's Dr. Dan, and
Right.
We have our community, and we're not doing too bad,
Yeah.
as far as a community replacement. But yeah, that's important.
Yeah.
And the other things that are important about spirituality, or about religion, is it gives you a feeling of peace of mind and connectedness to your body and the universe. And that is all something you can have without religion. You don't need the dogma.
Yeah. And I think a more, I don't know, realistic, fulfilling way. And if you're comfortable deluding yourself, I suppose the other way can be fine. But if you have that doubt, I don't find that to be fulfilling. And but there are ways of doing it for sure. I totally agree. Yep. And that, like, you can have a spiritualistic atheism. Absolutely. Yeah. I agree 100%.
Now, the thing with dogma and religion is, like, a religious person will imagine an atheist as being a thing without their dogma, which is controlling how they behave. And then if they imagine you without the dogma, they're saying, well, you've got to be some kind of nihilistic monster because you have no guiding dogma. Well, we are guided by what we are as organisms, as what we are as primates. We are social creatures who want to hug each other. We want to snuggle up and, together in the cold nights, and protect each other and raise our children. I mean, we have all of these qualities that just come from the nature of what we are as organisms.
Exactly. Right? Yeah.
And that when you dogmatize that, you're in danger of creating a dogma that will run into a situation where you won't come up with the right answer because now you've applied dogma to the thing rather than
and then it can lead to this moralizing over things like, say, drug addiction, alcohol addiction, mental illness, where it's not a moral issue at all. It's a medical issue. And
that drives me crazy. Basically the judgment of alcoholics and drug addicts being one, having gone through that
I've had family,
that horrible razor, and looking out at people that are still suffering, and to see people judging them, and not having willpower or being somehow morally
lack of character, and it's just awful. We know that there are specific brain networks that are very affected by drugs and alcohol. And addiction is a real thing, and that it's manifested in the brain. We know a ton about this, and that's the thing that's so frustrating. We've known about this. I mean, it's been worked on for 60, 70 years in great detail of the brain circuits that are involved, and we only learn more and more about how mechanistic it is, and that it isn't an issue of character or moral failing at all. And it's just sad that
or lack of willpower. Like willpower in an alcoholic has been commandeered to go out and get more alcohol.
Yes, exactly.
And alcohol, and the other thing about among recovering alcoholics, there's it's kind of common knowledge that our morals are perhaps too finely tuned. Even though we constantly break them when we drink, we hate ourselves more than anybody else because of it. Because we are very
deep-seated shame. Yeah,
we have this deep-seated, and the shame contributes to that circuit. That basically keeps the loop going for addiction. But I mean, it seems like we're not spending enough. I don't think we're researching all of this enough. I don't know. Maybe or at least it isn't filtering down, because we're still.
It might not be filtering down. There's research on it quite a bit. I mean, at least as far as the neuromechanisms of drug addiction, alcohol addiction. But I mean, there's still a lot we don't know. That's for sure. And the seeking treatments such as medication that could potentially help. I've got a colleague here at Virginia Tech whose specialty is figuring out what certain kind of orphan G protein coupled receptors do in the brain. And he studies this in the context of addiction. He's primarily working right now on nicotine addiction, but he's done other things and he's seeking, like, are there ways that we can manipulate these specific neurons that have certain kinds of G protein coupled receptors with certain pharmaceuticals that could potentially affect the pathway and break down the habit forming processes that have occurred that now just take over the brain, so that we can deal with the withdrawal symptoms and also the seeking behaviors. And I mean, that's just one aspect. So a lot of there's work being done, but it's slow work and it's hard. The brain's complicated, that's for sure. I mean, we're working hard on it, though. As if I can take the credit for their work, but I suppose I'll take a tiny little bit.
Wonderful if you could pop a pill and quit smoking. Or,
so that's what his hope is, that he'd be able to do that, and we'll see.
That was the founder of AA, Bill Wilson. That was his hope back in 1930, that they were that basically science was going to discover a drug that would just cure them, that sort of thing. And and here we are, what is that, 70, 90 years or something?
Right, and still waiting for it. That does not mean that it's a moral failing, though, or a lack of character or anything like that. It's very mechanistic. We know that for a fact. Absolutely.
Yeah, it's really fascinating. Like, I've always been a keeper of subjective experiences. So I've always, like, had this watcher that watches me, even when I was using. And I noticed things about myself, like when I was in a cycle of addiction, I would never be able to predict who I was going to be or what I was going to be thinking 3 days hence. So it was like I could feel myself being manipulated by something much more powerful than my thinky brain, right?
Sure.
That made me use again. So I would I would quit using. I would spend 3 days recovering, and then all of a sudden I'd be like, oh, I think I'll get a drink, to celebrate, right? And it's like, where the hell did that come from? And it was almost like it came from some deep subliminal portion of myself. And I realized that I was completely fucked, that there was nothing in the world that was going to keep me from going back into another cycle, right, and doing it again. And at the very end of my using, which was about 16 years ago, I was absolutely desperate. I couldn't believe, like, I would just, I mean, I had been brought to a low. Brought to a low that was just so incredibly physically and mentally wiped out that I could not quit. And my son asked me one time, I was crying and saying, I wanted to quit. I want to quit. I don't want to use anymore. And he said, well, why don't you just quit? And I said, Dylan, in 3 days, I'm going to be a different person. And I have no control over who that person is. I had no control over who I was going to be 3 days hence because of that addictive cycle. And that is the important thing to understand when you're dealing with an addict, is they are locked into some kind of
it's a loop,
a cyclic thing.
It is.
That is killing them.
Right.
And they need help. It's like
there's absolutely. There's the reward part of it, which is the reinforcing part, and that's the thing that'll get you started. But eventually you also have management of withdrawal symptoms. So that's going to be part of it. So, like, once you have stopped using, that's when withdrawal is going to occur. That negativity, so now you're going to try to avoid the negative symptoms of it. So that's going to then trigger some of the habit forming part that has been already established, if it's been reinforced enough, to then go out and find the reward. Then all of a sudden, your reward, you alleviate the withdrawal. But of course, then the consequences of the reward. And so then you stop using, and then you have the withdrawal, and it's a cycle. Absolutely. And there's a habit reinforcement part of it too. Involves the basal ganglia, in fact.
The reward part of it is probably the most misunderstood by non-addicts. The reward mostly happens while you're obtaining the drug, not after you've done it.
That's right.
Because when you're really sick, when you're really in the cycle, you are miserable, and the whole time, like, you'll start drinking, and you'll get to a point and you'll feel sick and you'll feel screwed up and you'll feel uncomfortable. So you will drink more and more and more, and pretty soon you're blacked out. But then when you look back on it, you realize that the most fun you had was when you were popping the first beer.
Right. Yep.
It wasn't afterwards. And that's what's incredibly sad and painful about it. When you look at somebody that's locked into that, is you realize that they are absolutely miserable. Right. They're not out there whooping it up and having fun. Nope. You know, they've gotten into a cycle of misery. But overall, I am so grateful that I am an alcoholic and a drug addict, and I got to go through all that.
Sure.
Because I understand it from the inside.
Right.
And I would never trade that experience for anything. I'd never go back.
Has an appreciation without going through the experience. They really should, though, right? And I think, and that's where this moralistic garbage feeds into that. It gives people that space to think that that's how it works. It has to do with character or morals, and it's not at all.
Yeah, you can have true compassion for a person that goes through it if you've been through it.
Yes, that's certainly true. But I think the fact that others struggle with it is because they have this goofy notion that it has something to do with character, and that's something that's not rooted in basic neurobiology when it is, and that especially the addiction is very much. We know the neural circuits. We know how the basics of this works, and even a lot of the details. And like that, how can you not see that as a clinical manifestation of a brain illness, basically?
Yeah. There's this treatment facility that started in Minneapolis, I think, called Team Spirit. And they basically believe that you use because you sin. And you have to get right with Jesus, and you have to basically list all your sins, and you have to admit that you're a sinner. And it's just a horrifying treatment program.
It's awful. Yeah. I mean, it's bad enough that Alcoholics Anonymous has the you got to believe in God thing, right? That sounds terrible.
Well, in Minnesota, it's that God thing is pretty underplayed, so it isn't as bad in some states as it is in others.
Right.
There's down south, it's a little different, I think. I hear, but anyway, we get around it. You know, we got a bunch of atheists that have a group. That's pretty cool. Anyway, okay, so for what are we at? Hour and a half?
About an hour and a half. Yeah, I do need to get going relatively soon. Did you want to touch on anything else?
No,
we started to talk about the protein heritable. I mean, I think it's so simplistic. It's
basically that's bullshit. And the other thing that's bullshit is, how did this evolve? You know, is what they keep coming up with. Right. Like he has a problem with.
It's God of the gaps. It's irreducible complexity is just complex incredulity is all that is. It's another. It's a way of just putting up something and saying, "Hey, you need to come up with some way of explaining this." Of course, evolution, and this is the thing about the protein world and RNA world. There's a lot of work that's being done on the abiogenesis front, but you know, obviously, no one was there to see what happened. Between 4 billion years ago to up to about 1 billion years ago. That's a long time for things to change, but all of it was happening at the single cellular level. So we don't have a fossil record of that. So of course that's going to be, hypothesized and somewhat, okay, let's just try to figure it out with what we can do in the lab. And I think we have great models, but I'm willing to concede that that doesn't mean that it worked this way. It just means that we have a reasonable way of saying this is one way that this can come about, and we can test these things and come up with various hypotheses and test them. To point to topoisomerase or the bacterial flagellum and say we have no explanation for this, therefore God did it. That's I don't know. It's incredulity. It's just the God of the gaps argument. It's a bunch of bullshit.
Yeah, my favorite thing is when Saul Cordova does a video. And he'll come up with he came up with mammary glands. How did those things evolve? And he said something like, so the animals started licking the mother's chest or something, and then milk formed or whatever. So what happens is, okay, I shut the video off and I go and download about 50 papers on how did this evolve. And then I spend about 2 months just studying that. And I have the greatest time ever.
Mhm.
But the thing that they never do is go and Google, how did this evolve,
No.
right? That's all you
Right.
got to do. Like if you Google, how did eukaryotes evolve? Paper after, thousands of papers on it, right? Mammary glands, limbs, fins to
Yeah.
limbs, all of it.
You can find
We
just
have
about
so
anything.
much.
And, you know, the thing is, he did show a slide from a paper that included a figure from a paper talking about evolution of topoisomerase. So he, of course, and I mean, this is the kind of his thing
Right.
right now. So of course he knows about these papers.
Yeah.
Um, but I looked a little bit at some of the topoisomerase papers. I mean, it's not my field of expertise, so I can't comment
Right.
on a great level of confidence. But it seems like reasonable explanations for how topoisomerase came about. And I mean, he just kind of like, oh, it's just a mess, and just skipped right over it. And I'm like,
Yeah. He showed the paper that
hmm,
basically
right.
talked about there's
And there's
two, I
several,
think
of
there's
course. That
two
that was a
different,
good review that went into
right.
detail about the pathway
Yeah.
that it could come about, and
Right.
laying out some of the evidence of what we know already that validates some of the evolution of the topoisomerase system. But it's like, okay, yeah, you're just skipping over stuff that people have already worked on, and so he
Yeah.
knows. It's not that he doesn't know.
Yeah. And there's a hypothesis, I think, that goes back to basically viruses
Mhm.
need to cut that DNA. So they need to glom on and cut it.
Right.
So and what you're looking at is you're looking at very, very small genomes in prebiotic life, right?
Mhm.
You had much smaller genomes that did not twist up, right, and they could
Right.
replicate. Okay,
Yes.
and then by the time and then you got viruses co-developing, right, and they're getting into the cell with their DNA, and they have things that can cut that DNA, and then genomes can get longer because now we have a thing that can cut that DNA.
Right. Yep.
And so it all makes a great deal of sense.
Sure.
And if you look at topoisomerase, I think there's two different
There's two different
types.
kinds. There's
Yeah,
topoisomerase 1 and topoisomerase 2. Yep.
yeah, and they seem like they're it was basically convergent evolution.
Yeah,
You know,
because they're very different. Yeah.
very different, right?
Right.
But then when you look at the mechanics of what happens, uh, the actual basically enzymatic activity of a protein. Okay, there are many different ways to skin one cat.
Right.
They're and they don't have to be that damn complicated to start
No.
with. They end up
Exactly.
looking complicated because domains add on and
Right.
things get more complex over the last billion years.
Right.
Like after eukaryotes, we basically had this explosion in complexity and mechanisms in proteins and
Right.
in biochemistry, basically,
Mhm.
that took off. But prior to that, there were simpler things, and you
Right.
got to go back and you got to look at it. And the thing is that what they have to prove is that it couldn't have happened.
Yeah.
And good luck with that.
That's right. Yes,
Because
exactly.
if you look at the complexity of biological chemistry and carbon chemistry, I mean, the complexity just is just astronomical.
And they operate on this assumption that whatever you're looking at the moment, this one, say, sequence of a particular topoisomerase, this is somehow that's like specially created, irreducibly complex. And it's clearly not, because if you compare the topoisomerase of a human to a mouse or human to a bird, like you're going to see substitutions here and there that clearly fall into this nested hierarchy of taxonomy, follows exactly what you would predict if there's just like slow evolution that occurs, and that you're going to have a mutation here and there, and it mostly is fine because the system then adapts to whatever subtle changes that have occurred in birds, say, versus in mammals. And it just continues to work, and that, I don't know, like, clear, if it was so special and that it was clearly designed, shouldn't it be identical in all animals? I don't
In all
understand
animals,
why
right?
you have every single protein that you look at, when you compare it to humans to chimps, they're almost identical, if not
Yeah.
completely identical. But when you compare it to humans to, say, birds, or humans to frogs, there are many differences. Some are very similar, but none of them are ever going to be completely identical, but many of them are very different, but they still do the same thing and they still act the same way. But with subtle differences, little tiny differences that, it's like, oh my God. But then you compare any one frog to another frog. Now those are basically identical, right? And we have 5,000 different species of frogs we can look at, and they're basically all the same.
Yeah.
I mean, come on. What is the explanation here that somehow that you have a creator that's specially designing the topoisomerase and frogs and making sure they all look the same, but then there's maybe a different one doing topoisomerase and mammals or just the apes, right? And then then when they started to work on the mice and the rats, well, they made a few mistakes and come on, like.
Well, I describe it as God had a dull pencil and he made all these mistakes as he was building the organisms or whatever.
My argument is that we don't have to assume that it's a single designer. Honestly, like I think that's one big lack in intelligent design is that they have this built-in assumption that it's a single designer, and we give them far too much leeway when we say that when we use the God word and when we say, I think that we have to nail them down, that like you cannot assume that it's a single designer. You look at all of life on Earth. How can you not think that there are multiple designers? It only makes sense if there's multiple designers. It does not make sense that there's a single designer creating snakes and mice, and that they want all the snakes to eat the mice, and that the parasite that lives inside the gut of a snake is designed by the same guy who designed the snake. And come on. Yeah,
I heard you bring that up. I never heard that particular angle before.
And so back in the day when I was a graduate student, I was pretty heavily, I would spend too much of my time on Access Research Network's discussion forum. Access Research Network was this kind of clearinghouse. It was supposed to be a clearinghouse for like research in intelligent design. And I think it was run by the Discovery Institute. I'm not entirely sure, but I actually know Sal from that. Like Sal was actually heavily involved in posting on there, but we also had William Dembski. He'd come on and post, and a lot of us would also like argue with them and fight with them over these stupid ideas. But there was one guy who came up with this idea of the multiple designer theory, this idea that we're like, there's a lack of effort in the intelligent design community to identify when, how, and who would do these things. And if you look at, we can use the analogy of human design. You can look at human designs and see that there are multiple designers. It's obvious. You can look for signatures of that design, right? But even just from the appearance of these designs, they compete with each other. If you look at the military systems of Russia versus the United States, clearly like there's similarities, but they're not the same, and that they compete, and they're in very direct ways, obviously. The same thing is in nature. It's the same thing. And how can you say that like it's a single designer? Because we know that human designs don't have a single designer. And we can see the traces of the fact that there's not a single designer in humans. There's lots of designers.
And then if you look at biochemical pathways, you've got a molecule that attaches to basically a membrane protein, and that signals something to happen with ATP, and that fires off and turns on another protein, and that protein inhibits another protein that inhibits another protein that ends up activating another protein, and then the whole thing goes into the nucleus and shuts off a piece of DNA. And it's like, wait a minute, why didn't you just go from the signal directly to the fucking nucleus?
So much of it, Rube Goldbergian.
Yeah, there's like it's 3 knots in a row, right?
Yes.
Which
right.
It's like, oh my God.
And that it's all ridiculously networked, where one protein might get phosphorylated, but it like I'm thinking of ERT-MAP kinase, where like there's phosphorylation event, and it leads to changes in all kinds of pathways, and there's some elegance in that, but it's also just super messy. Because now you don't have a lot of control over those things, and it's just because of, like we were talking about, you get these random associations that get locked in, but some of it gets borrowed and used in different pathways, and it's enough to drive something. But why, it's so ludicrously complex, I feel like,
Yeah.
that it does not look like the sign of intelligence to me, that's for sure,
And
because
if you
how
look,
ludicrously complex it is.
if you look at the interaction of a protein, basically you look at the connectome for any protein,
Mhm.
some of them interact with another 100 different things,
Sure.
right?
Yeah.
And out of those things, we know about, maybe one or two that
Right.
are valid pathways that we can figure out. But there's this huge network of interaction going on, and it's so obvious that there are many paths through there that
Yes.
can lead to either beneficial or deleterious or just plain neutral evolution.
Sure. Absolutely.
And,
And the neutral's a big part of it too.
and that's kind of underappreciated as to, they want to talk about how these proteins are so specifically designed to lock and key into each
Right.
other. No,
No,
they
it's definitely
connect with dozens of other proteins,
sure.
and they interact and stuff.
Right.
Anyway, we
Yeah.
should wrap up so you can get
Yeah.
going.
Yeah.
But,
Hey, this has been
so
fun. Yes. Let's keep in touch.
yes,
I we'll see how, if I get invited on to do more debates. I might be touching on a debate with transgenderism next time, which is an issue I've gotten more interested in, and I can argue it from a neuroscience perspective, which is something that
good.
hasn't been
Good.
touched on for modern day debates. And I think that, I don't know, we'll see how persuasive
That
it is.
one
But
I would love to see touched on, basically, because that's a big hot button. Okay, and then if
Cool.
you have any specific little "how did this evolve" problem, reach out to me.
Sure.
I've got papers.
We'll do.
Okay, so anyway,
Good deal.
I'm going to end the recording. So
Thanks, Pete. All right. Take care.